Hello. In several lectures, we have seen about the structure of DNA, how it was deciphered. And at the end, it was Watson and Crick using all the information from whatever is present and also putting in their own thought and tried and has actually successfully built a theoretical model which was later proved experimentally that is the case. So what we know as a quick review, it is the bases that are present, ATGC, and then the nitrogenous bases, then deoxyribose in the case of DNA, and the phosphate. And from Chargaff's rules, they know that it is ATGC. And from, the composition or the proportion of A is always equal to that of T, and then the proportion of G is equal to that of C. And from Maurice Wilkins and Rosalind Franklin's X-ray data, they figured that it is helical in nature, and then it has repeating subunits, and also that the structure is actually uniform in diameter, kind of. They used many different logics, and also one of the logics that we will look at, why should there be A should be base paired with T and G should be base paired with cytosine. So basically, when we look into it, the biggest contribution probably in my view is finding out this hydrogen base pairing and then the hydrogen bonds between them. Base pairing, so that and hydrogen bonds, the differences will determine the inheritance of that information. So one of the logics that they have used is the nitrogenous bases. The purines have two rings like this, and pyrimidines have one ring. So if we were to put two purines together, say for example this is adenine and this is guanine, then the diameter, the space occupied would be too much. If we were to put two pyrimidines, something like this is thymine and this is cytosine, then there are two near, or this is an empty space. Only when we have one purine and one pyrimidine, then we have this space absolutely spaced in this case like this. And that is one another logic. And I think when they were building the model, they must have found that this is how the distance, if they were to form the hydrogen bonds, they should have been within this close, within a certain distance. And that is one of the logics they have used. And we also should recollect that they are anti-parallel, 5' and 3'. The complementary strand would be 5' to 3'. And then the base pairing is, of course, as already described, A base pairs with T and G base pairs with C. 2 hydrogen bonds with AT base pair, 3 hydrogen bonds between GC base pair. And the backbone is on the outside. We have a deoxyribose, a phosphate, deoxyribose, phosphate, deoxyribose and phosphate. We have alternating molecules there. I am Dr. BCMR, Assistant Professor at Shastra. The structure of DNA has inspired numerous modern art and the sculptures across the globe, and these are some of the examples beautifully made and as a piece of art, and that is where science meets the art. I mean, this is also, we can keep on thinking about that the origins of these have started from that of the models built by Watson and Crick. There are even structures built for school children or for kindergarten children to play around with the structure, and this must be probably 2 hydrogen bonds, so it might be an AT base pair. It would be wonderful to play around, and while doing so also try to learn about the structure of DNA. So the intricate details and some of the terminologies is what we are supposed to learn, and this is a structure of DNA and I'm using this to discuss what exactly is going to be the minor groove and major groove. The place between these two, this is, each one of it is a strand of DNA. They are base pair or they are together. And here we have one strand and we have another one, and we also have this space here and there is a space here. This is called as the minor groove, and the space, the large space here, is called as the major groove. And why these become important is because DNA is interacted upon by many different types of proteins. And as we progress in the course, we will also discuss, we'll find out how many different types of proteins are there in regulation, replication, every way. If we see, they are interacted upon by proteins. And the interaction should happen based on the functional groups or reactive groups that are present on the DNA and also with that of the proteins. So if we see what kind of functional groups are exposed in minor groove and major groove, we can see some pattern in there. So these are the exposed groups on the major groove. On here we have the minor groove. So hydrogen bond acceptors are indicated as A, hydrogen bond donors are indicated as D, and nonpolar hydrogens as H, and methyl groups as M. So we are only looking at the exposed groups with which the proteins can interact. So on the major groove side, these are some of the groups that are there for GC base pair: A, and D, and H. If we have the GC and if we have it as a CG base pair, we would have the reverse of it, AADH, right, or HDAA. If you look for AT and TA, we also have some difference, that is ADAM, something like Adam. And if it were TA base pair, then we have MATA. So one of the differences that we can think of when it comes to the differences between what functional groups are available on the major groove and the minor groove. In the minor groove, if you look at it, say the CG and GC and CG base pairs. For CG, it is ADA. Those are the functional groups that are available here. And the same way, if it is CG, we would still have ADA. So if any protein is interacting at that particular location, it cannot make out whether it is a GC base pair or CG base pair from the minor groove. But if you look at the major groove, we have a difference. AADH, if it is a GC base pair, and it is a HDAA if it is a CG base pair. Similarly, we can do the same for AT and TA base pairs. On the major groove, there is a difference. That means proteins have an opportunity to distinguish AT base pair from that of TA base pair. Whereas on the minor groove, there is not much information, because AHA in either case, if it is AT or TA, it is just AHA. So this becomes important to say when we discuss about the interactions between minor groove and major groove. I mean the protein interactions with the DNA. Majority of the interactions, especially those of sequence specific proteins that bind to DNA, would have to make major interactions from the major groove, because they can read, almost it's like they can read the sequence and through interactions. There are several other DNA forms, say something like aDNA, bDNA, and zDNA or zDNA. So they do differ in several features. One of it, ssDNA, the way I remember, is short and stumpy, and the ssDNA is long and slender. What I mean is, if something is too short, it tries to widen up. That is what is ssDNA. And ssDNA is on the reverse. When you stretch it, it is little thinner. What is in between? Sorry, this is ssDNA. What is in between is bDNA. That is, majority of the DNA in majority of the cells is in the B form. Z and A can form in different conditions. There are certain other changes. It's not just about the length, but there are also other changes, in which case we will focus on a few of them, and then we'll try to understand. The first one is handedness of helix. So here I'll try to demonstrate what is a handedness. I have a measuring cylinder and I have an aluminum foil, and I'm trying to now fold it, twist it around this measuring cylinder to demonstrate what is a left-handed. So I'm holding it with my left hand. This is the direction of my left hand holding it, and this is the direction towards the upward. This is the direction of the turn, and this is the direction in which I'm going. So I'll hold it this way in my left hand fingers, something like this, and I'm twisting it around the measuring cylinder in the direction of these fingers. I would go like this. There we have a left-handed, what is meant by left-handed coil. That is a left-handed coil. You can see that this is the direction of forward movement, this is the direction of the twist. You can turn it upside down and it would still maintain the same thing. The turn is in the direction of my left hand and it is towards the forward. This is how I'm looking at. I hope you can see that. So to distinguish it from left-handed, let's also make a right-handed one. So I'm holding it in my right hand. This is the direction of my fingers, and here is the direction in which I'm going to twist forward. So, and I'm going to twist it in the direction of my fingers, these fingers. And I'm doing it. And there we have a right-handed helix or a twister. Here we have it, a right-handed helix. So you can see that the turn is made in the direction of my fingers, and it is being turned towards the up like this, if I'm visualizing it the same way. Let's compare what is the difference between a left-handed helix and that of a right-handed helix. I hope you can see that their designs are, the way they turn are towards this way, like this. And I can put them upside down and they still are in the same right-handedness or left-handedness. Majority of the DNA, especially the ones that we're talking of, ssDNA is the left, A and B DNA are right-handed helices. This is the right-handed helix. And whereas the the ssDNA is a left-handed one. And then the repeating units are for A and B, they are one base pair, whereas in ssDNA it is two base pairs. If you see the sequence, it is usually rich in CG, and they do form, this is a kind of one repeating unit in each case, and that forms the left-handed DNA. Another important factor that we will remember is the number of bases per turn, and it is about 10.5 approximately for that of bDNA. But many times when we are doing calculations based on these, sometimes we'll just call it as 10 turns 10 base pairs per turn, whereas zDNA has 12 bases because it's the longest one. And the others, we will also discuss a few more. That is, before going in there, we will also see that because of zDNA and that of bDNA, the minor groove has changes. This is the minor groove in case of the bDNA, and all these exposed area is the major groove, whereas in zDNA we have it in a different way. So there is something we will have to remember. One turn is about 3.4 nanometers or 34 angstroms, and number of bases, as I said, it's usually 10.5 base pairs per turn, but for some calculations for ease of calculations, we use 10 base pairs as well per turn. So if somebody has given us the length of DNA is, say, for example, 10 to the power 4 millimeters, and then they could ask you to calculate the number of turns or calculate the number of base pairs that DNA has. And those are some of the interesting kind of problems that usually come up. If you can, please pause the video here and you might want to try it out by yourselves. There is one more important thing that we need to discuss is about the two words denaturation and degradation. What do they mean when we say denaturation and degradation? So we have two strands here, and these strands, say this is a double-stranded DNA. I'm just putting it linear, and here we have the base pairs, the hydrogen bonds between these two strands. So if this DNA becomes, there are two important types of bonds that we need to remember that hold the DNA together. One of it is the hydrogen bonds, and the second one, or which we should have been the first one, is to make these strands is the phosphodiester bonds, or phosphate backbone, we'll just call it that way. So phosphodiester bonds make up the bonds here, and the hydrogen bonds are indicated as blue ones here. So if a DNA molecule has denatured, they would have become single strands like this. That is called as denaturation. So in this case, the bonds that were broken are the hydrogen bonds. If we had a DNA molecule which could have become, where there is a breakage of the phosphodiester bonds like this double-stranded DNA molecule is broken down into two pieces by breaking of the phosphodiester bond, then we call it as degradation. So in degradation, the phosphodiester bonds are broken. In denaturation, it is the hydrogen bonds that are broken. The denaturation is highly reversible. If you put these two complementary strands together, they can base pair again and form a double-strand DNA based on the conditions around. So the process of forming a double-stranded by two complementary DNA molecules is called as renaturation. So I hope we can remember these words, and that is denaturation, breakage of hydrogen bonds. The reverse of denaturation, that is two complementary single-strand DNA molecules forming one double-strand DNA, is renaturation. And there is degradation, which is not a reversible one, and that is breakage of the phosphodiester bonds in the phosphate backbone of the strands. Thank you very much.